All right, so in this video, we're talking about proteins. So protein is a word I think is used very loosely in society. People say protein for a lot of things, but what exactly is a protein? Well, first of all, as it says here, proteins make up greater than 50% of an organism's dry weight or biomass. The names of most proteins end in lin, while the names of most enzymes, which are composed of proteins, usually end in ase. So a way that I like to think about enzymes are proteins that do work. All right, and so if you see a word that ends with ase, you know we're talking about an enzyme. If you see a word that ends with an N or a lin, there's a protein, like penicillin, amoxicillin. What are those? Those are antibiotics. If you get an infection, you go to the doctor. The doctor may prescribe you an antibiotic, and it may end in a lin. So you know that antibiotics are proteins. Proteins are large macromolecules, which are composed of monomers or building blocks called amino acids. There are 20 different amino acids used by living things to make proteins. Proteins and enzymes usually have hundreds to thousands of amino acids in their structure. All right, so what is an amino acid? An amino acid has 4 different parts. Okay, so you have an amine N, or an NH2. That's where you get that amino acid. This N can act as a base by accepting a hydrogen ion, and then you have a carboxyl end, which is a COOH, which is on the other end. This part acts as an acid because it can release a hydrogen ion. So we have one end that accepts a hydrogen ion, and one can release a hydrogen ion. All right, so every amino acid has an alpha carbon. This is the central carbon atom to which all the other functional groups are attached. And then we have an R group. This part is the only part that is different in the 20 different amino acids. The R group gives different amino acids their distinctive properties. So when we change the R group, we change the structure, thus we change its function. Structure equals function, just like when we were talking about all the other molecules, talking about the carbohydrates, flipping that hydroxide ion, turning the glucose, alpha glucose to a beta glucose. One gives you a starch, one gives you cellulose. Just like steroids, changing those functional groups, structure equals function. All right, so let's take a look at a basic amino acid structure. So right here is the central, the alpha carbon. It is the main man. All the other parts are bound to this alpha carbon. All right, and here is just a hydrogen atom. But here's your amino group, the NH2. Here's your R group, and here is your carboxyl group, your COOH. Now remember, one N donates a hydrogen ion, and one N accepts a hydrogen ion. So if we look down here, amino acid number 1, amino acid number 2. So here are 2 monomers that are going to bind together to form a polymer. Now, specifically, these monomers are called amino acids. Okay, and they're going to form a peptide, a dipeptide. So look right here. This one is your amine group is the end that donates a hydrogen. It's going to donate it to this hydroxide ion that's going to accept it. And so when that happens, you form a peptide bond, which is a type of covalent bond. So then your amine group of this one will bind to the carbon of the carboxyl group on this end, thus forming your dipeptide and releasing water. So what type of reaction just occurred? Well, we lost the water, and when you lose the water, you dehydrate. And then whenever you make something bigger or something new, you synthesize. So a dehydration synthesis reaction is what we just created there. So some key words here I just said to point out: peptide bonds are covalent bonds between amino acids. As we saw here, these are called peptide bonds. So when we put, so this is a dipeptide. If we put, let's say, 10 of these together, what do we then have? What prefix means many? We then have a polypeptide, right? So, and then we talked about the dehydration synthesis. So this is a dehydration synthesis. So let's say we wanted to break these apart. What type of reaction would we use? Hydrolysis. So then we would use a water to break that bond. And that does happen in proteins, but more often they are put together to make new proteins. All right, moving on. So here we go again. Here's another diagram. Here's one amino acid. Here's a second amino acid. Notice the amine end of one is donating a proton to the carboxyl end that is accepting. When I say proton, I mean hydrogen. Anytime I say proton, I mean hydrogen, because hydrogen is number 1 on the periodic table. Therefore, it is almost synonymous to say it is a proton, because it has one proton. Is basically what it is. All it is a proton. So this amine group donates a hydrogen ion. This carboxyl group accepts the hydrogen ion or proton, and then forms this nice, strong peptide bond, which is a type of covalent bond. All right. All right. So what all of this information tells you is that the amino acids, because of the way that they're bound together in the structure, the polypeptide chain, which is when you link a bunch of these together, has directionality, meaning one end functions for something that the other end does not function for. It's not like chaining a bunch of glucose molecules together, where one end has the same function as the other end. Here, this end we have a amino end, and this end we have a carboxyl end. So the amino end is called the N-terminus. And the other end, the carboxyl end, is called the C-terminus. So something can happen on this end, donating a proton, that cannot happen on this end, which would be accepting a proton. So this has directionality. And that's very important, because these amino acids will be arranged in a specific order, which we'll talk about more in just a minute. Okay, so let's go ahead and get into levels of protein structure. All right, so as the amino acids are linked together, so let's get the baby toys back out here. All right, so here's my baby toys. So I used this earlier for your visual for monomers forming polymers. So one monomer bonds to more monomers, and we get a polymer. Now in this case, one monomer specifically is what? An amino acid. All right, one end of the amino acid can accept hydrogen, and the other end donates hydrogen. I really like these things. They're very simple, but such a great visual. All right, so what end accepts hydrogen? COOH, the carboxyl end, or the C-terminus. And so then this end would be what? The amino end, or the N-terminus. So we have what type of reaction? We lose water and form a bigger molecule, so we have a dehydration synthesis. All right, so when you link enough of these together, you form a linear structure, a line, basically. And so this is known as our primary structure. So look right here. Primary structure refers to the sequence of bonded amino acids The primary structure of each protein is coded for by DNA. A mutation in DNA can change the primary structure of a protein. This change can vastly change the overall shape of the protein and cause it to lose its function. All right, so we have here a pink, a purple, orange, and a blue. So let's say we have a mutation in the DNA, and instead of coding for a purple here, it codes for another blue. So remember, structure equals function. So now we just changed our structure. So the function of this protein is also going to change. No longer is this one purple, it is blue. The blue one doesn't do the same thing as the purple one. All right, so that's very important to understand. That's how mutations work. And from the primary structure, we go into the secondary structure. This refers to the shape of the specific sections of each polypeptide chain. Okay, now we have two types of secondary structures. So go down, we have an image. Here's the first shape. We could have what's called an alpha helix. So a helix should make you think of a spiral-shaped structure. And then we can have a pleated sheet. So here we have the primary structure is lined up and then folded and wrapped around, and then folded again and wrapped around. All right, so for example, here's my primary structure. It is linear. Now, due to the characteristics of each amino acid, what can happen is I can either form an alpha helix. So if I just wrap this around my hand, like so, not knock everything down, we start to form an alpha helix. All right, so what causes that alpha helix? Well, the function of the amino acids, but look right here. These dots, what type of bond is represented by dots instead of a solid line? Remember, it's just a weak attraction between hydrogen and another element, so hydrogen bonds. So hydrogen bond forms here and forms there and there and there, and causes this structure to spiral, spin, and form this helix. Now, in the case of a pleated sheet, we have almost like making a sandwich. All right, so we're folding this up like this, folding it up like this. I don't know if this is how you guys make sandwiches, but, you know. Basically, I'm trying to say that you're stacking them up like this right here. All right, so this would be a pleated sheet. And what causes this to happen? Well, if you look here, we have hydrogen bonds causing this pleated sheet to occur. All right, so for proteins that are built by ribosomes located in the rough ER, this folding takes place within the rough endoplasmic reticulum. Hopefully you remember from early on that within the cell, you have a rough endoplasmic reticulum and a smooth endoplasmic reticulum. So what does the rough code for? Well, it helps to fold these proteins into their secondary structures. That might be a question you see in the future. I would probably guess that it is going to be, like, definitely on the quiz. Where does the secondary structure fold? Where does that action occur? Well, it occurs in the rough endoplasmic reticulum. Then these proteins are usually exported from the cell through the process of exocytosis or secretion. Other proteins are constructed by ribosomes which float freely inside of all. These proteins typically remain in the cell where they are made. The folding of these proteins usually takes place within a chaperonin. Chaperonins are a protective structure, which, by the way, it ends with an IN, so it's probably made out of proteins. They're protective structures that allow proteins to fold without water present. So it gives the proteins a nice, safe place to fold. And we'll have to look more at chaperonins later. I'm running out of time.